Uh, Last week, before a week ago, we did with photosynthesis. Photosynthesis. So photosynthesis we did with light reactions and dark reactions. What is the purpose of light and dark reaction? The purpose is to synthesize, is a plant resource or using the light energy into chemical energy or food or glucose. In other words, what we did, we prepare, okay, we prepare our food, meaning glucose we did, or starch synthesis, right, by photosynthesis, by the plant. But today what we are going to do, we are going to study how to manage the food what we eat. So mainly we are going to study today about the glucose, how the glucose is stored in our body, and how the glucose is being used in our body. This one, how it is used, which already we have done, the glycolysis, TCA cycle to produce the energy. But this time we are going to study about the storage part of that one. So we don't have the photosynthesis capacity, but we can eat glucose or sucrose or whatever the plant products or starch, and we are going to manage how we are going to utilize our very schematic way, meaning very economical way. If you use unnecessarily, you will spend a lot of glucose, then you become tired, and you're not eating much of food or glucose, then you are going to starve, and also the consequences, and also how this storage of glucose or utilization of this particular food product like glucose polymer and glycogen. We are going to study about the glycogen, how the glucose is stored in our body as a glycogen, and that's the metabolism we are going to study. That is on the glucose storage. What is the glucose storage in our body? How? And then how they are from storehouse, from stored glucose to useful for our function, for our body. Okay. So this is the main theme or goal or whatever you call it, our main objective of today's topic. Okay. In the textbook we see the runners, the sports persons are running there, and mainly they are using the stored energy in muscle, and that's the part is the glycogen. Glycogen, Glycogen. Glycogen is the polymer of glucose. So whatever you starch or polysaccharide, whatever if you eat, that is from the plant derived product of this one, and that is going to split into monosaccharide and absorb and getting into the blood, and then you are going to have excess of the glucose, and that is going to be stored as a polymer in the tissue. What is polymer again? If I draw the structure of the glucose like this, just coming out like a ring structure of glucose, like 1, 2, 3, 4, 5, 6 carbon units on that, and now if you have, this is 1 unit of glucose, and this is another unit of glucose. So I'll draw like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like, you know, it's just one. So one round is the one glucose molecule, like that, like this one. So they are linked with each other and to become a polymer. Okay. Now, the glucose is a precursor of many molecules. Okay. Glucose is the one glycogen, but glucose is the precursor, P-R-E-C-U-R, precursor for the biosynthesis of many molecules, many molecules. So glucose is the precursor for the biosynthesis of many molecules. So meaning the glucose is absolutely necessary for our body function and produced for the other compounds. Now, how the glucose is getting into the tissue? By you feeding your food or glucose or in the form of starch and other carbohydrates, whatever you eat, that is being broken down to glucose ultimately. Now, the body cannot store as a glucose. That is the problem. Okay, that meaning glucose cannot be stored as glucose molecule. Why? The reason is, if the cells, okay, the body cells, individual cells, if they accumulate glucose, what will happen? It will absorb water molecule and the cell will burst. Meaning, if you have excess amount of glucose inside the cell, it will give you, the cells an osmotic shock. So you cannot store the glucose inside the cell. In other words, the glucose can be stored as a polymer, like one glucose molecule, like a long chain of this molecule, and this molecule as just like a granule, they call it granules, and these granules cannot attract no water, and it as a substance, as a particle. So you get, suppose if this is a cell, if this is an animal cell nucleus, if these dots are a granule, it will not, because each one will have several chains of polymer of glucose, and it cannot attract more of water. Rather than if there is a one single molecule of glucose, it can attract. That meaning the size of the molecule is a polymer, larger size. It acts like a particle, and it cannot absorb any water. But if you have an individual monomer, just a single molecule, if you have more single molecules are there individually, it can act like an osmotic shock. It will absorb more of water and the cell will burst. So by favorable for the body to store glucose, it should be in the state of a granule or polymer. So polymer of glucose is favored. Okay, so that's what just I want to mention to you. Now, this polymer can withstand, okay, in osmotic, or there is no non-osmotically stored. Also, this polymer in need, if you need more of glucose, what will happen? This glycogen molecule can release what? Release glucose whenever there is a need to the cell. So that's what is the advantage. So we need to know what is the metabolism of this important molecule, glycogen. Okay, that's what the chapter, what we are going to study today. What is the type of molecule is glycogen? It is very, very large molecule. As I mentioned earlier, it's also it is a branched one. It's a branched, okay, branched polymer. What is branched polymer? If I write like this, a glucose molecule, another glucose, it's a straight line, right? It goes like this. But the branched one will go like this: 1, 2, 3, 4, 5. Like, you know, it goes like about 10, and then you have another molecule of glucose chain polymer, and then after 10, another one, another branch will start. So what you have, this is the straight chain of one, but this is the glycogen will have a branched polymer, very large as a branched polymer. Okay. And whenever there is a need, this branched one or this glucose can be broken down, and then that is released for the energy purpose. Okay. How long this glycogen molecule? How long? As I said, it is a polymer, but how long it is, and it has like a 12 layers because of the branch, you know, one to the other, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, like that. You know, all of them are branching from one to the other, and then so you can have 12 layers of this particular polymer of glucose, and the length or the thickness is approximately 40 nanometer thickness of that granule. So how this molecule is linked with each other, glucose molecule is linked with each other, how they make a polymer. So now let's go for the structure of glucose, like just write on lecture form. Okay. Say 1, 2, 3, 4, 5, 6, like, okay. Carbon, I'm just writing the ring structure of the glucose molecule. Okay. Here they used to go like this is the first one, and what have the next one? What was it? This is another molecule of glucose. Yeah, this is a polymer, right? So you know about it. So this is one molecule, this is a second molecule. So there is the first carbon and then the fourth carbon. They are linked with a bond, a glycosidic linkage. So glucose, this is another glucose. This glucose, this attached together, and this is formed because first carbon and the fourth carbon of the next glucose molecule. So it is called 1, 4, 1 or arrow, or the people get a 1, or 4 glycosidic linkage. So 1, 4 glycosidic linkage. It forms what this is the first carbon of this. This is the fourth carbon of this molecule. So this will go chain with the another one. So this is the second one. So this is the 1, 4 linkage. So what will happen here? 1, 4 linkage. If it grows like this, 1, 4 linkage as a straight line, like a line, or it grows in a one direction. Just I'll write it another paper. Yeah, one direction or straight polymer. Okay. However, there is a linkage at the 6th position. That is, I'll write it here. This is the 1, 4 linkage, 1, 4 linkage, 1, 4 linkage. Okay, it comes out like that. So when this particular glucose molecule, which is just I'm drawing a picture here, like this. I can also check into the textbook as well. I'll just go through. So here, 1, 2, 3, 4, 5, 6th one here, 6th one, and this is going to be linked with the first carbon of the another one which is coming up. So the 1, 4 which is going like this, and then the sixth one which is starting from here, and this sixth one will link with the first carbon for the other one, another one like this. This is the next one. So this is the first carbon of the another chain. So here you have the next chain will grow. So this is the sixth carbon and this is the first carbon. I'll write it in a different way now, so that the branch alone I am writing it. So you will understand that one here. So this is the sixth one, and this is going to alpha 4. This is the next parallel sideways, or this is the fourth, this is the first one. And what happened here, the sixth carbon over here, the CH1 here, CH2O, and here the glycosidic linkage will go on this, and the first one of the other one, okay, first carbon of the next one here, which is going on to, and then you have a 1,4 linkage. 1,4 linkage which is going over there. So it goes over, I will show into the figure, probably that will help. So the sixth carbon which is going to the other side, and then the first carbon of the next chain. So you have the branch is alpha 1,6 linkage. So you should know about the alpha 1,4 linkage, that is on the straight line growth of the chain, and the alpha 1,6 linkage is only on the branch of the glucose molecule, where it will have the link over that one. Okay. So in short, I will write it like this: 1, 2, 3, 4, and then you have a sixth one will start like this, and then 1,4 like this, and then the 1,6 here goes like this, and then if this is the 1,6 linkage, this is 1,6, or wherever the 1,6 linkage which is coming out, that is a branch, and then always the horizontal one which is growing over the side, that is a alpha 1,4 glycosidic bond. So in this way, the glucose is stored as a polymer, as a straight chain as well as the branched one, and thereby it can grow. The glycogen molecule is stored. Okay. And there is a pattern on this of the branch. The branch start every 10th residue. So it is not like everything is going to be a few, and then after 5 or another 6 or another 10 or 20, another branch start. No. Every time there is a 10th residue. For example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, there's a branch. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, there's a branch. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10th residue, there's a branch. So it grows like this, and then there's a 10th. So it grows like this, and then a 10th. So every 10th residue, you get a branch of 1,6 linkage which is coming up. So that you should understand that now. Okay. Now, this is, I mentioned before, alpha linkages. Alpha linkages. And this alpha linkages always show as a helical, like a DNA double helical like this structure, right? You know about the DNA double helical go on like this. So this particular shape of the polymer also in a helical pattern, helical structure in the alpha linkage. The another one, there is a beta linkage which is also there, and the beta linkage, and this beta linkage mainly present in cellulose as a straight chain, as the beta linkages. So that's why the sweet, whatever you eat, or the starch you can digest, you can chew, the amylase can react on that, alpha amylases. But you cannot chew your cloth or paper. Paper is made up of cellulose, right? So the linkage of the glucose, both of them are made of the glucose. Glucose is made up of cellulose also, composition of cellulose also glucose. But the linkage of bond from one glucose, this is another glucose, this is another glucose. The linkage between here and this, each molecule, the linkage is beta linkage. This you cannot digest. You don't have enzyme for the beta linkage. So that's why Unless otherwise you have a cellulase enzyme that is only in bacteria, in some fungi, as well as in some other, other plant. You have a beta linkage of enzyme, that is cellulase enzyme. We don't have a cellulase enzyme, that we have to depend upon our own bacteria. So bacteria in our system, or intestinal cells, intestinal where they are living, they produce more of cellulase enzyme. So that will cleave this glucose and release some energy. So we don't have this one. So that's why we cannot eat cloth or eat of a paper or anything like. Otherwise, anybody can eat this paper like a sandwich, right? So it's not possible. That's the beta linkage. Okay. So it forms like a straight fiber. That's what the cellulose fibers will do. Fibers. That's beta linkage. So coming to our point, it's a glycogen, and that is not reduced. Glycogen is stored. I said it's a storehouse of glucose molecule. Okay. But what happened in the fatty acids? Fatty acids, that is also in an adipose tissue, that's also storage house, also is an energy rich. When the fatty acid, they oxidize, which we are going to see in future in a couple of classes, the fatty acid oxidation, where it will produce, in presence of oxygen, it produces ATP and energy storage house. Okay. But in the glycogen, which will also produce more of glucose, and then glucose is entering in the metabolism, and then we are getting much of the energy or ATP. But compared to the fatty acid, glycogen is a bit different, because glycogen is stored, okay, for our food, for brain. Because normal between the food, if we eat morning breakfast to the lunch, and in between that the brain needs the fuel. In between that the liver that store the glycogen, which will release small quantity of glucose, so that our blood glucose remain constant, or managing the blood glucose level, or homeostasis of blood glucose will be maintained because of the glycogen that is stored in the liver. So that's what we are going to see how that is going to happen. Why? The brain, as I mentioned earlier, only fuel. The brain will use only the fuel from glucose. Unless otherwise, if there is a starvation, there is a chance the brain cells, or the neuroglial cells of the brain, they may use from fatty acid like ketone bodies. So they can use the ketones as one of the energy fuel. That's only for the prolonged starvation, not for 1 hour or 2 hour or one time without meal. No, it's not. But if you starve for 24 hours, then your blood ketone levels will go up. So that ketone can be used by the brain as a fuel to give the energy. Okay. Unlike the fatty acid, which needs the oxygen to release the ATP, as I mentioned earlier, fatty acid needs oxygen. Okay, they need oxygen, and that will release, that responsible for producing more of ATP. But the released glucose can produce the glucose from glycogen. They can produce ATP without oxygen. So that is the neat part. So if you have a fatty acid, you need more of respiratory oxygen, then only you have an energy level. But glucose is there. ATP can be synthesized without oxygen by anaerobic, in the absence of, by anaerobic activity. Okay. Now we will see where the glycogen is stored, the storehouse or warehouse or the storage. Though all cells, they have a little bit of glycogen, all cells and the tissues, they have glycogen, little bit, small quantity glycogen. Okay, small quantity. But mostly liver and muscle. They have more of stored glycogen. Among the liver and muscle, liver will have more. Liver has more glycogen than muscle. Okay. Now, liver, how much that more means? Liver has 10% and muscle the 2%, and the skeletal muscle mass, they have an amount of when you have 100 gram of liver, the 10 gram of that particular liver is glycogen. And muscle, if you have 100 gram of muscle, if you take it, 2 gram you can get the glycogen. So that is the quantity which they are storing inside the cells of the liver and muscle. And they are, where they present in the cells? They present in cytoplasm. Cytoplasm as granules. It's just like a salt granule. If you see in the chemistry, a small particle, which is staying on that. You can also check into your textbook 21.3, which I'll show you a bit later. And the liver, now we will see one by one. Okay, what happened in the liver? It will produce like a liver glycogen synthesis. Glycogen synthesis is going on in the liver. Okay, and glycogen breakdown is also going on. Glycogen breakdown. So what do you mean by metabolism? What we studied in studies of intermediary metabolism. The metabolism is the anabolism and catabolism. The anabolism is the synthesis and catabolism is the breakdown. So we are studying today about the glycogen breakdown as well as the glycogen synthesis, and the organ mainly on the liver, and we are also seeing in the muscle as well today. And mainly on muscle, the glycogen, is a breaking down or breakdown, which we are studying there. The muscle. And whenever there is a need of a muscle energy, then it will break down and then get the energy from the glucose. But in the liver, you have glycogen synthesis and the glycogen breakdown, both occur. But when the liver breaks down into the glucose molecule, that's applied to the whole body. I'll write it separately. When the liver, which will break down the glycogen, and then the glucose used whole body need through what? Through blood. So it gives the glucose into the bloodstream, and the blood supply to all the parts of the body, and thereby the liver participates. Whereas in the muscle, the stored glycogen will produce glucose, but used only on muscle, not the whole body. So that you should understand. There is a difference between the functional difference between the liver and muscle. Liver is more of donor to the whole system, where the muscle is specific for a particular function. Okay, and only for muscle. Muscle glycogen is not available for the other body parts. Now, if the glycogen is broken down, it produces an intermediate, like glycogen is breaking down, it produces an intermediate called glucose 6-phosphate, because it's breaking down to glucose, and the glucose on one molecule of glucose, and that converted to glucose 6-phosphate, And this glucose 6-phosphate is derived from glycogen as an aerobic or anaerobic metabolism. And in the liver, the glucose 6-phosphate is converted into liver to glucose, okay, and then to blood. That's what I mentioned earlier. And to be processed, in other words, it's also processed to generate glucose 6-phosphate, NADPH. Otherwise, we also studied last class of the pentose phosphate. Okay. So in other words, the whole glycogen metabolism, and they need to regulate. You know, you cannot have all the, when you have more of liver, when you produce more of the blood glucose, when you have to stop the blood glucose release, and when do you need of NADPH need, and when do you want to stop it. All this, you know, the activity of the glycogen breaking down is being done by or regulated by hormones. Regulated by hormones. We will see what hormones and how do they play. Okay. And this process of glycogen breakdown requires, of several enzyme processes. Several, not one enzyme, several enzymes. We are going to see those enzymes and their regulation. Okay. Have you got any doubt or any question so far? Cinco Ranch? Victoria? Are you following?
Yes.
Yes. Yes. Okay, great. Now, the phosphorylase, we are going to study the enzymes that is involved in glycogen metabolism. We are going to say specifically few enzymes. Just to follow through, it is very simple and it is easier. Okay, don't get confused. Enzymes, the enzymes, first enzyme you should remember is phosphorylase, PHO, yes, PH, phospho, PHO, or YLASE, phosphorylase enzyme. This is one enzyme. It catalyzes the phosphorylytic LYTIC, phosphorylytic cleavage of glycogen to release glucose, okay, 1-phosphate. First step. There is a glycogen molecule, as I mentioned earlier, glycogen, there's a polymer, and there's a branch as well, right? So there is a position. It will not go and cleave this glucose or this glucose or this. And there is a specific region, whichever the periphery of this non-reducing end of, I'll show you, non-reducing end of this molecule, the periphery, the last one, the external one, okay, and that is going to be attacked by a phosphorylase enzyme, and then it will remove one unit of that, and thereby the rest of the molecule is the -1, okay. In other words, I put it like this in a equation, glycogen, okay, we call it N residue. I'll write it in a separate paper. Glycogen, N means total number, you know. So you call it in any, suppose you have 100 or 1000, you know, N become any number, glycogen molecule, plus and in a phosphate molecule, okay. And what the product is? Glucose 1-phosphate, because you are adding the phosphate to a glycogen. This is a polymer. These have any number, plus a glycogen molecule, glycogen in a mathematical expression n minus 1, because this 1 is coming over there, n minus 1 glycogen molecule, and the enzyme is catalyzed by the phosphorylase enzyme here. So just remember, we are not using ATP, we are using an inorganic phosphate here, and that is very important here. Okay, so this one will go on to the next phosphorylase enzyme reaction for this, and then from this another molecule of glucose 1-phosphate will be produced, and you get what from the total n minus 2, and then n minus 3, and then n minus 4. Like that, each cycle you get more of glucose 1-phosphate is produced, and slowly the glycogen is going to be reduced to minus 1, minus 1, minus 1. Where they will attack on the glycogen molecule, as I mentioned, it will attack the glycogen polymer at non-reducing end of glycogen polymer. That is, what is non-reducing end? The ends with a free hydroxyl group. Free hydroxyl group. Wherever there is a free hydroxyl group on the end, that is the position where that is at the fourth carbon of the glucose molecule, which is present at the periphery. So the fourth carbon where there is a hydroxyl group, and that is the one we call it as a non-reducing end of glycogen molecule, where the phosphorylase enzyme will bind, and then the orthophosphate, they cleave that particular bond between the C1, that the carbon 1, and the terminal is the C4 is adjacent. As I mentioned earlier, suppose this is the one, and this is another molecule as the fourth carbon. This is the first carbon, so it will go and cleave over there. So what you get really on the periphery, that's the reducing end of that particular 1,4 cleavage. Okay. Now you have the product from the phosphorylase. What you have the product? Glucose 1-phosphate, meaning the first carbon is phosphorylated by the inorganic phosphate. Then what will happen? This glucose 1-phosphate is being converted to glucose 6-phosphate, and the glucose 6-phosphate, then, as I mentioned earlier, it is converted into glucose in a different pathway. But glucose 1-phosphate to the glucose 6-phosphate is the second step in glycogen breakdown. Okay, this process is done by phosphoglucomutase. Okay, the enzyme called phosphoglucomutase. One word. Okay, I'll write it separately. Glucose 1-phosphate. This is the product of glycogen, first one, and then now you get the glucose 6-phosphate molecule. The first carbon, which is attached to the phosphate, is position is changed to the same molecule at the 6th position, and the enzyme is catalyzed by phosphoglucomutase enzyme. Okay, so this we are going to see how this mechanism is occurring here. Okay, a bit later. The mechanism of this phosphorylase enzyme, how this phosphorylase enzyme is acting to come to this product. So that is interesting. One, the phosphorylase enzyme. Now I'm talking about phosphorylase enzyme. That's the first step. Okay, phosphorylase enzyme. It is a homodimer. Homodimer, meaning it has this two enzyme subunit. One unit, this is another unit. Both of them are identical, and in your textbook you can also verify figure 21.5, and you have one is the yellow color and the another one is the white in color, and they have both of them, they have a site, a catalytic site or active site and that the enzyme that is called pyridoxal phosphate. Pyridoxal phosphate. That is the one which is the catalytic site, pyridoxal phosphate, where the glucose or glycogen molecule or the terminal non-reducing end is binding to it, and then that will produce the glucose 1-phosphate. So the pyridoxal phosphate, it is a unique process and it replaces the ATP. So there is no ATP for this reaction, phosphorylation reaction, and the ATP for this ATP is saved. Why? Saved for other glucose or glycolytic glucose breakdown. You remember earlier in glycolysis, for glucose breakdown, we added ATP molecules and then coming down to pyruvate and TCA cycle. So we need ATP. So for the stored glycogen that is broken down into individual glucose, the ATP molecule is not necessary. There is no need. So that's the bottom point. So phosphorylases never uses ATP for glycogen breakdown. Also, there is another one which I mentioned earlier, alpha 1,4 linkage, that is being done by the phosphorylase. Okay, it is breaking down. But you remember that you have a branching point, alpha 1,6 linkage at the branch, at the branching point. In the branch, you have, the unique feature is the alpha 1,4 linkage up to, it will read up to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So what happened? The phosphorylase will activate to 1,4 linkage here down, and this has been removed, and this is removed, and this is removed, and this is removed, and this is removed. At to this 6 residues it can act. But what happened? The 4 residues over there, the phosphorylase cannot act on this one, because the 4th residue you have a branch of 1/6 branch there, linkage is there. So as soon as it reaches the 6th residue, yes, it will come easily, but the 4th residue before the branching one, it cannot, the phosphorylase cannot act on that one. At that time, what happened here, there's a transferase enzyme will act. Okay, as I mentioned earlier, like this, it will coming out, and then it'll all on a sudden a branch, it will come out like this. Okay, so the transferase enzyme, as soon as you remove these, all of them, the transferase enzyme will break down this part and then move to this one, to the same other branch, like alpha 1,4 linkage. It comes out like this. So the product looks like this. So because the new one which is coming over here is the new one, and this has been transferred, this particular residue to over here, and then you have only one molecule of 1,6 linkage, and this is being easily cleaved by an enzyme. So there are 2 enzymes. One is the transferase, transfer the residue. This is called transferase enzyme, which will transfer the rest of the residue on the 1/6 branch to the side chain, and the another one, the alpha 1,6 glycosidase enzyme, which will cleave this, and thereby it releases the 1 molecule of glucose. And then what happened here, when you add, the phosphorylase will go here, 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 goes on cutting, and then finally all the glycogen molecule is being broken down. So that is the debranching enzyme. Okay. So if you remember the additional two enzyme apart from phosphorylase here, 2 enzymes are alpha 1,6 glucosidase enzyme for the branch, and then a transferase enzyme, transferase. So how many we have studied? Previously we did study phosphorylase enzyme 1 and then 2, okay, 3 enzymes to break down the polymer of the glycogen. And they are also called remodel the glycogen molecule. This one, because of the transferase, they remodel glycogen molecule, glycogen. so that it can easily being attacked by the phosphorylase enzyme, and you will have more of a glucose 6 phosphate, glucose 1 phosphate and then glucose 6 phosphate from this one. So the next large step is, as I mentioned earlier, like a glucose 1 phosphate to glucose 6 phosphate enzyme. And the enzyme, what is called, we did study before, phosphoglucomutase enzyme. Okay, in your textbook, it's a figure 21.9 that we give on that. And this one, the active mutase enzyme contains a phosphorylated serine molecule. So this is very important to know about the position of phosphate group. Move from first carbon to sixth carbon. Okay. So it is not the same phosphate move from first carbon to sixth carbon. That's what I want to tell you about it. Okay. The phosphate which is going to the sixth carbon is not the first carbon phosphate group. So the enzyme which contains the phosphorylated serine, molecule, serine or serine, and this is a hydroxyl amino acid, which you remember, amino acid. It present in the active site of the enzyme, of phosphoglucomutase, and this hydroxyl group, you have a phosphate group is attached already there. So the phosphorylated serine presented in the active site of the phosphoglucomutase, that phosphate is being transferred to the sixth position. I'll just explain into the picture. That's very easy to understand now. So the first carbon phosphate is shuttled to serine. Then what happened? The first carbon which is present in the glucose, first carbon phosphate from glucose will go to serine, because the serine already donated to the phosphate to the sixth carbon of the glucose molecule. So the serine is empty now, so it received the first carbon phosphate to it. So in thereby you get the phosphoglucomutase will act from glucose 1 phosphate to glucose 6 phosphate. Okay, now what you have now? Glucose 6 phosphate by the enzyme phosphoglucomutase from glucose 1 phosphate to glucose 6 phosphate. Now in the liver, there is an enzyme called glucose 6 phosphatase. This is like a scissor. The phosphatase which will cleave this phosphate group out from the glucose molecule, and thereby it releases the glucose 6 phosphate. It releases the glucose molecule, and the phosphate molecule separately. The phosphatase means ATASE. Always remember, phosphatase means if you remove phosphate. It is the opposite to phosphorylase enzyme, the first enzyme which we started, phosphorylase, which will add the phosphate to the glycogen molecule. That is the phosphorylase enzyme. And phosphatase is just opposite. Because in biomedical sciences, the phosphorylase and phosphokinase enzyme, which will add the phosphate group, and phosphatase, they may play a major role in disease prevention and other pathogenesis of diseases. So we are going to see a little bit of the regulation of the phosphorylases and the phosphatase mechanism after a break. So I'll give you a 10-minute break, come back, and we will follow it up rest of the class. Okay? Do you have any questions? The phosphorylase is regulated by allosteric interactions. So that's what we want to study now. Phosphorylase, that's the enzyme which we studied, right? Phosphorylase enzyme. And could you see document camera? Phosphorylase enzyme. Regulation. regulation by allosteric interaction. How many of you studied biochemistry of allosteric? Otherwise, I will just refresh your memory. Allosteric interactions. Allosteric interaction. Again, I'll write it here. Interactions. I don't know whether I can see clearly. Allosteric interactions. I will change my pen. Allosteric means, suppose if this is the enzyme, okay, and your substrate is binding over here, and normally you get a product like a different color of this product, and you get the enzyme again being released. So what happens in an allosteric means if apart from the active side, this is the active side, apart from the active side, some other compound or the inhibitors regulatory molecule will bind the other than the active site, and thereby it may induce the reaction or it may suppress this reaction. So allosterically activated means this enzyme may have another compound which will facilitate this reaction, so you have a more of this reaction or the more of this activity is being favored. In other words, in some other compound, that may bind with in a different positions or the same position over here, and that will lead to inhibition of this. If you have more compound, will more of inhibition. So that is called allosteric inhibition. So you should remember in biochemistry, the enzyme and their active site is the specific for binding of the substrate. So this compound is not is going to bind in the active site. So it leaves the active site open. However, they might either inhibit or it might activate this enzyme. So if they are activating, then it is called allosteric activation. And if it is inhibiting, means they are called allosteric inhibition. In other words, in a different context, if the same one, suppose if this compound, which I mentioned here, if this compound which is binding into the active site, that meaning it binds competitively to the substrate. The black color is the substrate, and this green one is the activator. So, or the inhibitor. Instead of binding here, if it binds over here, and it competitively it inhibit. This compound may inhibit competitively. It may inhibit competitively because the active site binding the substrate is going to be occupied by this inhibitor competitively. Allosterically means it might, apart from the active site, it may bind some other place, and thereby it may activate or inhibit. So these are some of the basic mechanism which we are going to call it as allosteric interactions and reversible phosphorylations. Okay, we are going to see how this phosphorylase enzyme is going to be regulated. So that's our primary. And these phosphorylase enzyme, and they are responsive to hormones. Another one, responsive to hormones. What hormones? Such as insulin, okay, epinephrine, and glucagon, glucagon. Okay. They have a different control mechanism in liver as well as in the muscle. Insulin, epinephrine, and glucagon. Okay. I also mentioned before the glycogen is stored in liver as well as in the muscle. So the mechanism, the different control mechanism, different control or regulatory mechanism is present in muscle as well as in liver. Okay. let us see in one of the example which I give you, a muscle phosphorylase. So as I mentioned earlier, glycogen present in the muscle, and the enzyme which act on that muscle glycogen is called muscle phosphorylase. Muscle phosphorylase enzyme. And this is being regulated by intracellular energy charge. So intracellular energy charge which will manipulate this muscle phosphorylase, meaning whenever there is a muscular contraction, or if you think that you have to contract your muscle, there is some changes is going on. Just for thinking, just for ready to in action, you are going to do some work, just think and act. So when you think something before you act, the muscle phosphorylase enzyme is being activated through the neuronal changes or the nervous system changes, through muscular and neurojunction changes, neurotransmitter changes. The energy charge is going to be altered, and thereby the muscle phosphorylase is going to be activated. Now, let us see what is the muscle phosphorylase. Let me call it as a phosphorylase. It exists as a two forms. As I mentioned earlier, the muscle phosphorylase activated means already there is an inactive form of this particular phosphorylase enzyme. Okay, let us see the two forms now. We are calling as phosphorylase A. We call it as a phosphorylase A, and this is an active form. I call it like active form of the phosphorylase A. The another one is inactive. Inactive. It is not having any action. Inactive form, that is phosphorylase B. This is an inactive form. So they form there are two type of state they can exist. The A, which is existing in a thermodynamically, they call it as a relaxed state. The chemical structure is much more relaxed one. Otherwise, we call it as R state. R state, relaxed state. Okay. The another type of state, which is the phosphorylase B, okay, which is a less active, but it is a tense one. So otherwise, it is called the T state. So R state and a T state. So if I say T state means the phosphorylase B, and that is an inactive form. If I say R state means they are relaxed form, but they are more of active phosphorylase. So there is an interconversion or changing or conversion of the T state to R state. In other words, the inactive form of phosphorylase to active form of phosphorylase. So this is the conversion, the changing. Okay, that you should understand. So the phosphorylase regulation means the inactive form of phosphorylase B is going to be active phosphorylase A, or from a T state into R state. Now, I mentioned earlier the inactive form of phosphorylase is going to be active form. How it is being activated? Now, the next question. How the inactive form will be activated? There is an enzyme called phosphorylase kinase enzyme. This phosphorylase kinase enzyme, okay, this is activated by this enzyme. This kinase is activated by calcium ions, okay, and phosphorylation. If there is a phosphorylation, a phosphate group is added to this enzyme, that's called phosphorylation. Okay, calcium and a phosphate group, phosphorylation, will activate the phosphorylase kinase enzyme. why I am calling about phosphorylase kinase? This enzyme, phosphorylase kinase, kinase means it could be, again, it's used on the ATP molecule, but the phosphorylase kinase is going to activate phosphorylase B. That's the inactive form, which I mentioned earlier. I'll show you. This is the inactive form of phosphorylase B, right? And then it will be converted to phosphorylase A, because the phosphorylase A is going to activate the glycogen molecule. I mean, binding with the glycogen molecule or the glucose molecule to convert to glucose 1-phosphate. I mean, that's the conversion at phosphorylase A. But to give this active form, we have to activate the inactive form, and this inactive form to active form, we need an enzyme. That is an enzyme called phosphorylase kinase. But in other words, this enzyme also need an activation by calcium ion and a phosphorylation reaction. Okay. Now we are going to see the phosphorylase kinase. About what is the phosphorylase kinase? This phosphorylase kinase, phosphorylase kinase, this enzyme has a 4 subunit: alpha, beta, okay, gamma, and delta. So there are 4 subunits. 4 subunit. Okay. In this 4 subunit of the phosphorylase kinase, as I mentioned earlier, it needs, as a calcium ion, it binds to one of the subunit. A phosphate group, it will bind to one of the subunit, and thereby the 4 subunit together will act on phosphorylase enzyme to activate into another form. So now we will see the hormones, okay, the hormones that will activate the beta subunits. Okay, phosphorylates the beta subunits. I'll tell you here. Hormones, okay, that phosphorylates the beta subunit of phosphorylase kinase enzyme. That's very important. Okay, and the calcium ion, the hormones phosphorylate beta subunit, calcium ions binding to delta subunit. So when these hormones and calcium, it will activate the phosphorylase kinase, and the phosphorylase kinase now activate the phosphorylase B enzyme, and then that will become phosphorylase A will be activated. The final destination is B is inactive form of phosphorylase, now become an active form of phosphorylase A. Okay. The next step, what type of hormones, which I mentioned earlier, hormones phosphorylate beta subunits. Now we will study the role of hormones. What hormones which will activate? Hormones, epinephrine and glucagon enzyme, and both of them are signal. The hormones are signal the need for glycogen breakdown. Do you make sense now? As I mentioned earlier, phosphorylase A, which is necessary for the glycogen breakdown. How the phosphorylase A is being activated? By B, from inactive form of phosphorylase B to active form. How the B is activated? Phosphorylase kinase enzyme. The phosphorylase kinase is how they activated? That's by hormone that phosphorylate or the beta subunit of that enzyme, and the calcium ion which will binding to delta subunit, and thereby it is being activated. Now what type of hormones will activate? Epinephrine and glucagon. In other words, epinephrine and glucagon, they signal the need for glycogen breaking down. In other words, and the epinephrine and the glucagon, they are the starving hormones. Whenever there is a starve, you don't get more of glucose. If you don't eat, the glucagon level will go up, and that will activate, the beta subunit phosphorylation, and calcium ion to delta subunit, and then the next step is to activate the phosphorylase kinase to phosphorylase B to A. And then glycogen will be eventually breaking down. So now we will see how the phosphorylase kinase is being activated, which I mentioned earlier too. Another enzyme we call the protein kinase. Protein kinase A. This also activates phosphorylase kinase along with the hormone and calcium. Phosphorylase kinase enzyme. So you remember protein kinase A, which will activate phosphorylase kinase enzyme. Okay, and the phosphorylase kinase, which will activate a phosphorylase A form to B form to A form. Glycogen breakdown enzyme. This is the enzyme which is ultimately we recovered. This should be activated. So protein kinase A, phosphorylase kinase, and then B, phosphorylase B, and then B to A. Okay, and the next step, what activates the protein kinase A? As I mentioned earlier, protein kinase A. Okay, the protein kinase A, which is activating the phosphorylase kinase, right? So the protein kinase A is being activated by G proteins. G proteins. Okay, G proteins. And if you recollect last week, Nobel Prize for the G protein coupled receptors, GPLC, that's got a Nobel Prize for that guy, Lefkowitz from Duke University. Okay, so the G protein, we were awaiting his, he will receive a Nobel Prize, we thought of 10 years before. Nobody recognized him at the time. But now more and more evidence is coming out, like a G protein, we know that G protein is acting, but he didn't receive Nobel Prize because that's a very good molecule, and also more of the drug companies are making more of drugs based upon G protein coupled receptors, but he didn't receive any Nobel Prize, but it's extensive work. But now it's been recognized, and G proteins, that make a sense this time. Okay, good. And the G protein transmit the signal. Okay, transmit the signal for initiation of glycogen. I put GLYN, glycogen breakdown. Say glycogen breakdown. Okay, protein kinase A through G protein. The G protein we will see now. Glucagon and epinephrine, as I mentioned before, glucagon and epinephrine. Epinephrine what? They activate the trigger. There's a trigger, the breakdown. Of glycogen. So you always remember glucagon and epinephrine. I put short form epi. How they do that one? Okay, in the muscular activity. So if you have a muscular activity, extensive of muscular activity, or anticipation of muscular activity. Anticipation. That means you think that you are going to work. You think that you are going to run. So those things are, the anticipation of muscular activity. Muscular activity. And that leads to release of what? Epinephrine. What is epinephrine? Epinephrine, in other words, it is called adrenaline. Have you ever? Adrenaline. See, there's some of the thrilling movies and everything, they call it adrenaline pumped up or something like that. They used to do a lot of adrenaline, So that kind of stuff which you think and you get fear and everything, the adrenaline is pumping out. This adrenaline, which is a catecholamine derived, catecholamine, an amine, catecholamine derived from tyrosine molecule. Tyrosine amino acid. It's an amino acid derivative, tyrosine molecule, and they are coming from adrenal medulla. Adrenal medulla. Otherwise, it's a stress hormone, right? a fear or stress, or if you have more of stress, the adrenal become, is getting secrete more of adrenaline, and that's why it is through the adrenal dependent, certain drugs which will activate the adrenal and thereby it suppress. So it is an adrenal dependent, adrenal activation. In other words, the anticipation that leads to epinephrine or adrenaline release, and thereby, they're breaking down of the muscular glycogen molecule. And also the epinephrine or the adrenaline, they stimulate glycogen in a muscle and not in liver. So it will, the adrenaline activate or the epinephrine activate the glycogen breakdown in muscle, not in liver. That's very important. Don't write liver glycogen breaking down by adrenal medulla or adrenaline. No, it is not. Okay. So muscular glycogen is broken down by adrenaline or epinephrine. So that's very important to remember. Now, the liver glycogen is more responsible by glucagon. Glucagon, this hormone, will act on liver storage of glycogen and releases glucose molecule. And the glucagon is a hormone, which is a polypeptide hormone. It is not like catecholamine, but this is a polypeptide, protein hormone. Polypeptide hormone. Okay. And glucagon is synthesized by which cell? You know about the pancreas of alpha cells or islets of, beta cells of Langerhans, or like here you got alpha cells of pancreas, which will secrete glucagon hormone. Okay. When it will synthesize the glucagon, whenever there is a low blood sugar. Whenever there is a low blood sugar, what will happen? Glucagon is being activated, and thereby it induces the liver glycogen breakdown. Okay. In other words, physiologically speaking, glucagon is a marker for starvation. If you starve, you have more of glucagon synthesized in your body. Okay. Next one. Glycogen is synthesized and degraded by different pathways. Okay. Now we will see the glycogen synthesis in a different way, like synthesis of glycogen. I mentioned breaking down, now a synthesis. The synthesis of glycogen, they requires synthesis of glycogen. That is GLYN, glycogen. Requires uridine diphosphate. Uridine di means 2, phosphate, uridine diphosphate glucose. So one compound. In other words, I write it in a short form, UDPG, like UDP glucose. So I am talking now the synthesis of glycogen. The previous lecture which I gave it to you, there are two types of glycogen, liver glycogen and muscle glycogen. Muscle glycogen is broken down by the hormone, which I mentioned to the different steps in the enzyme, and the hormone is responsible for the breaking down, and the hormone is the initial step. The liver, glucagon, and the muscle, we did study about adrenaline or epinephrine. Now we are studying about the synthesis of glycogen, and this requires a molecule called uridine diphosphate glucose molecule, and thereby it will add more of glucose like that. So you have several glucose molecules are there. Okay, this molecule cannot form a polymer on its own. It cannot, or if an enzyme, nothing. But this molecule, this glucose molecule, individual glucose molecule, should be linked with UDP, like UDP. So all of them, uridine diphosphate is linked together When these molecules are there, then what will happen? The enzyme which will activate the joining. Otherwise, the glucose molecule, they are inactive molecules. These glucose molecules are not active. But as soon as the uridine diphosphate molecule is attached to the glucose, this is being so active, so it can be added to the, or it can used for the polymer synthesis or glycogen synthesis. So that's what I want you to remember now. But here, in a degradation process, the earlier one, what we studied, degradation process, it requires phosphate ion, like phosphate molecule, phosphate, direct phosphate. Say glucose, phosphorylase enzyme will be added, that the phosphate is being added to the glycogen molecule, and thereby glycogen is breaking down. But here, it's in a different way. UDP is being required as an energized molecule, UDP. In other words, the UDP glucose is an activated form of glucose. Yes, an activated. It's not sleeping, but it is busy. It is being activated glucose, meaning to synthesize more of glucose polymer. And it is something like you studied earlier, the ATP molecule is the energy currency, right? Or the normal phosphate, but if it is the ATP, it is an energy currency. And also acetyl-CoA, acetyl coenzyme A. And both of them are active forms of phosphate molecule. If I add phosphate, nothing will happen. But the same phosphate in the form of ATP, adenosine triphosphate, yes, that phosphate is so active. Or the phosphate is present in acetyl coenzyme A molecule, that's more effective. The same way, UDP will activate the glucose molecule, and thereby the glucose is being elevated or more active, so that will be used for the synthesis of the chain. So the active form is very important. Okay. Where they are active are the addition of the UDP. It is on the carbon 1 of the hydroxyl group of the glucose molecule, and also with a diphospho group. Diphospho, that's what we are calling as a UDP, diphospho group. And what happened, finally end, is this glucose is active. Otherwise, it is inactive. So, how this UDP glucose is being produced together? It forms from UTP, that's a uridine triphosphate. Like ATP, this is called UTP, uridine triphosphate, and glucose 1-phosphate. They react together, and the enzyme which will connect with these two molecules, which is called UDP glucose phosphorylase enzyme. UDP glucose phosphorylase enzyme. Okay, UDP glucose pyrophosphorylase enzyme. Pyro, meaning the product is going to be pyrophosphate. So UDP glucose pyrophosphate, PYROPHOS UDP glucose pyrophosphorylase enzyme will act on this one as an enzyme and together to form the substrate. So the product of this enzyme will be what? UDP glucose and plus a pyrophosphate, meaning double diphosphate. So that's why that enzyme called pyrophosphorylase enzyme. Okay. Now the enzyme, ultimately the enzyme which we call it as a glycogen synthase. Glycogen synthase is the enzyme which will link the UDP glucose and plus another UDP glucose, So it will add one by one to form a polymer. So the glycogen synthase is the enzyme which will act on the UDP glucose. But it will not act like as I mentioned before, when all these molecules are being activated and then all of a sudden the glycogen synthase molecule, suppose this is the red one is the glycogen synthase molecule, okay, this is the enzyme. So it will come everything together and then it will synthesize a polymer. No, it is not something like that. But before that glycogen synthase, it needs some sort of primer, okay. So the glycogen synthase can add, it can glycogen synthase. Synthase means synthesis, the glycogen synthesis. So the glycogen synthase is the enzyme. Glycogen synthase can add new glucose molecule, glucose, where there are 4 residues already there. Where there are 4 residues, glucose residues, meaning glucose, 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 glucose. So if whenever there is a 4 residues, a small chain of 4, then only the glycogen synthase will act on it. If it's individually, if it's not as a 4 unit, it won't react. It needs a 4 unit as a primer, as a substrate. Otherwise we call it as a primer. Like a DNA primer which you have done before in your earlier class in biochemistry and cell molecular biology for building up of the DNA for the PCR and other things, you know about, you heard of the primer. So like that here we need a 4 residue act as a primer. So primer to build on, okay. So the priming of the 4 units, 4 glycoside units, okay, that is called priming. And this priming is done by glycogenin, Glycogenin, glycogenin. This glycogenin, glucosyl transferase, otherwise we call it as this is an enzyme called glucosyl transferase enzyme, glucosyl transferase enzyme. This enzyme, glucogenin, glucosyl transferase, it will produce a short 4 residues. So when these 4 residues is formed, from where? From individual glucose molecule, it forms a short 4 residue. That's it. That's the only purpose of this particular enzyme. After that, the priming is done. The priming is the glycogenin. Then after this priming, what happens is, you have the glycogen synthase will use the UDP glucose molecule, build a glycogen molecule from this synthesis, which is going on and on and on. So that is the proper way to do that one. And the glycogen, every glycogen molecule, if you see the glycogen molecule in each and every molecule of the glycogen, you have a central portion of glycogenin like this, and then you get of more of the red one is a glycogenin, and that's the primer. And this primer, you get more of the glycogen molecule is being built on the chain. The polymer will grow from here. Okay. And a branching enzyme is also being involved, and that form from here, branching enzyme. Already the branching is 1-alpha 1,6 linkage. So the branching enzyme also involve that form 1,6 linkage, and thereby the glycogen is synthesis. So with this, I have given a brief outline of how the glycogen synthesis is taking place and how the glycogen is being broken down into individual glucose molecule. Now I want you to go through these PowerPoint, and where we will go on further on whatever we have done so far. I want to have a review, and then we will study a little bit further about some other diseases here, okay? Any doubts so far? So here, why the slide? The slide shows mainly on the runners. You have a more of muscle, and where you need glucose breaking down to produce energy, and muscle stored glycogen. That's it. And different step, epinephrine, the muscle, more of epinephrine, and that involves the phosphorylase, the protein kinase A, and then that activate the phosphorylase B to A, that the different steps involved, and then glycogen to glucose for energy is being there. It's going on the stepwise process. And here, this is the glycogen in the central portion, which is called as a primer. From this, everything is built on. So this is the glycogen polymer, glucose polymer, and glycogen as a stick, or the long one here with the branches and everything. And here you can see the non-reducing end on this both sides, and how this is the 1,6 linkage and 1,4. This is the 1,4 linkage, and this is the 1, and then the fourth one, 1 and 6th carbon linkage. So this is the branching point, 1,6 linkage, and this is the 1,4 linkage for the growth of the chain. Okay. And this is a glycogen granule in the electron micrograph. We can see the dark dots on that. That's the glycogen molecule which is stored inside the liver cell. And how this is the step glycogen in my N-1, and the glycogen phosphorylase, glucose 1-phosphate, and then the phosphoglucomutase enzyme, which I explained earlier. So that convert to glucose 6-phosphate in the muscle and brain. The glycolysis and liver, glucose 6-phosphate glucose into the blood, and then here in the muscle, the glucose and glycolysis, pyruvate convert to lactate and carbon dioxide and water, and then pyruvate into the TCA cycle. But it is being prevented because more of pyruvate is converted into lactic acid. That's why you have a muscular pain whenever you have a large amount of, you did the intensive exercise workout. And also glucose 6-phosphate is the source for the pentose phosphate, where the ribose and NADPH is being synthesized from this. So there are 3 ways the glucose 6-phosphate is being used. From where you are getting it? From glycogen, as well as glucose. Glycogen end residue, how the glycogen molecule is being broken down by phosphate. There is no ATPs involved here. So phosphorylase enzyme, here you can say, the glucose is being released here, and glucose of 1-phosphate, and this is a glycogen N-1 residue, because 1 residue is being released from the polymer. Okay, the first step in the breakdown of glycogen. And this one is the catalytic site, how the phosphorylase enzyme, you have 2 homodimer. This is one particular molecule, another molecule, and this cartoon shows where exactly the catalytic site is there for the binding of glycogen. So you can have the glycogen binding sites, and these are catalytic sites where the this is a nitrogen terminal and C terminal domain of this particular protein. And you can see this phosphate group, there lysine and then phospholipid glycine, binding site of the phosphate group or the substrate. Glycogen binding sites here and glycogen binding sites over there. This is the mechanism how the lysine they form the changes, how they donate the phosphate group in the carbocation intermediates and in the glycogen molecule. This is the mechanism. Glycogen residue alpha 1,6 glucosidase, because where the branching, the glycosidase enzyme cleave the 1,6 linkage, wherever the branch, that is being broken down. Okay. Now you can see the entire molecule, 1,6 linkage, 1,4 linkage, phosphorylase. You can see that one here, and transferase enzyme. Could you see this one? This is the branch. So this transferase, the blue molecule, which is being changed to here, over here. So from here, the branch is by transferase enzyme. And then the glucoside 1,6 linkage is being cleaved out by hydrolysis. So this glucose is released. And then this one is individually, it is being released just like this. It is being released. This all of the molecule. That's why this entire cartoon will show this stepwise process will removal of glucose from glycogen molecule. This is again how the one mutase reaction, like a phosphate, serine phosphate group, which is donate to 6th carbon, and then the first carbon is released back to the serine. Could you see the color? The pink will go here, and the blue will go back onto the serine molecule, and thereby glucose 1-phosphate is converted to glucose 6-phosphate. It is not the 6th position will go into the 1st position, no. It is being done by glucose 1,6-bisphosphate and then re-established to the serine. So serine receive a new phosphate from glucose. That's the understanding. And again, the phosphorylase A in R state and then the T state like a A and then B, and this is the cartoon how that will work. Then again, this is a different way you can express that particular mechanism of phosphorylase B to A, the active sites, and back to ATP and glucose molecule. You can just follow through the diagram. It's self-explanatory from one state into the another. And here you can see the hormones of the protein kinase A, like a phosphorylation here reaction. As I mentioned, there's a 4 subunit alpha, beta, and gamma and delta. You can see the inactive phosphorylase kinase become an active, and how this phosphorylase B to A is being changed from this. Okay. How the calcium ions being donated and where exactly the calcium is binding and phosphate is binding. You can see all these reactions over there, hormones and calciums involved in the activations of this enzyme. And this is the fasting, what happened, the low blood glucose, how that is being converted, and during exercise, and the muscle cell, how that glucose is being broken, glycogen is being broken down in both different pathways, and getting into the blood and the circulation. And this is the G protein coupled receptors, how we can see the GTP to adenylate cyclase enzyme, and this is the signaling kinase reaction with the protein kinase, phosphorylase kinase, and phosphorylase B to A, and all these, which is activated by epinephrine in muscle and glucagon in liver. So both share this GTP or G protein coupled receptor, G protein. So if you have a drug which will act on this G proteins, and that will inhibit or activate this process, that's why this guy, Dr. Lefkowitz, got Nobel Prize, and his student also got Nobel Prize from Stanford for elucidating the G protein pathway with the X-ray crystallography, where the GTP is binding onto this molecule. So that's why they got Nobel Prize for this year. Okay. Epinephrine, the molecule, and the glucagon molecule. That is the tyrosine derivative, and here catecholamiine, and here this is a polypeptide, hormone. Both of them are involved in glycogen breakdown. That's the thing. Okay. And uridine diphosphate glucose, UDP glucose, how this is the synthesis, this molecule. You can see the UDP structure, the pyrophosphate is attached to the glucose molecule. When this molecule is there, there is no activation. But as soon as it binds with the UDP, this glucose is highly active. That's the thing. Now, how this glucose 1-phosphate UTP, the formation of UDP glucose is the first step in synthesis of glycogen. First step, how the glucose is being activated. UDP glucose, uridine, glycogen, the N residue, and then N+1 residue, addition of uridine. And this is the growing 1,4 linkage, and then here the branching enzyme, that will give 1,6 linkage. So now we are synthesizing the glycogen by synthase extends of both non-reducing ends followed by more branching. So you have more and glycogen molecule is being produced. This is the G again. This is the one which I mentioned earlier, the glycogenin, where the 4 residues of the primer. From this, it will start building up more of branches of glycogen. This is during exercise or fasting what happens. This is the mechanism which you can see how this glycogen is being activated into the cell. And after a meal or rest, or the glucagon or epinephrine, switch on and switch off the mechanism whenever there is a need, So after a meal, it will store, and after a fasting or exercise, it is being released. Okay. And during exercise or fasting, again the same metabolism, and then how this binding region and genin and then how the inhibitors they activate and inactivate this process. Insulin, how the insulin will act. Insulin, if there is no insulin, what will happen? This reaction won't happen. and then phosphorylation of protein kinase, glycogen synthase to glycogen synthase, and glycogen, the glycogen synthase reaction, that's going to be more of glycogen synthesis process. If you have more insulin, that will induce the glucose to activate the glycogen synthase reaction, may be stored. So if there is no insulin, the glucose cannot be used to form glycogen, and the glucose will exist in the blood and increase the blood sugar value, and that induces diabetes condition. Okay, so less insulin. If more insulin, the glucose is being converted to glycogen. Here the phosphorylase A to B in minutes, and glucose is added. This is being activated. This is very interesting enzyme kinetics reaction, which you have studied in your biochemistry class earlier. And this is again another mechanism how this is being activated, glycogen phosphorylase A, B. Okay. And this some of the glycogen storage disease. This is very important as a biomedical perspective. Von Gilk disease, glucose 6-phosphatase or transport system is a defective enzyme in liver and kidney, increased in amount in glycogen in the affected organ, and clinical feature, massive enlargement of liver, failure to thrive, severe hypoglycemia, ketosis, hyperuricemia, and hyperlipemia. All of them are von Gilk disease. Pompe disease is more common in lysosomal storage, alpha-1,4-glycosidase enzyme defective. So what will happen in all organ, you have massive increased amount of normal structure of this. And then cardiorespiratory failure causes death, and usually before the age of 2. Pompe disease is not, it is lethal. And then Cori disease, third, that the amylo-1,6-glucosidase debranching enzyme, muscle and liver, and then increased amount short branch in like type 1 diabetes and this milder course. And then the Anderson disease and McCarthy disease, phosphorylase in the muscle, and then Hess disease in phosphorylase, and then seventh type is phosphofructokinase in muscle, increased amount of normal structure of the glycogen, and like type 5, that is a McCardle. And this is a phosphorylase kinase B. This is the 8th type of disease, glycogen storage disease. So glycogen storage disease is the 8 types of disease, and these are the affected organs and clinical features, which should be remembered now. And this is a lysosome, and when you see the glycogen is stored there, you can see the granules of the lysosomal storage disease. And then the ADP and rest and exercise with the normal and the heavy exercise, how much ADP is being produced in McCarthy disease and light exercise during the cramps, muscular cramps, and after acclimation to light and exercise. So these are some of the graphical representation of the ADP in micromolar concentration. Okay. And this is the kilodalton of sample 1, alpha amylase sample 1, and then sample 2 here, the alpha amylase molecular, polyacrylamide gel electrophoresis to determine the sample of the presence of the alpha amylase reaction. Alpha amylase 1, 2, 3, 4, kilodalton 66, you can see that one here. So with this, we are finishing this particular chapter 21. Okay. So do you have any questions so far? Any questions?